Hello, welcome again to King's Bioproduction Academy. Today we want to learn about the genetics. Let's discuss about genetics. What's genetics in definition? Genetics is the study of variation. It's the scientific study of variation. Inheritance and variation, sorry. Inheritance and what? We're speaking about inheritance and variation. Inheritance and variation. When we speak about inheritance, those are the structures that the offspring inherit from their parents. But when we speak about variation, those are about changes. There will be changes between the parents and the offspring. And what brings about the variation is the genetic makeup of the organism. This is because of the gametes. We gain some gametes from some of the parents. I mean, that's now the male and the female. The male gives at least an haploid, then the female gives the haploid half number of their chromosomal number to their offsprings, and that half number we get a full, which gives a diploid within the offsprings. So from these things again, we'll understand more of some things like the homozygosity and even the heterozygous. When we speak about the homozygous, we speak about the alleles being similar, but the heterozygous, we speak about the alleles being different. What are these alleles? These alleles are structures, they are the genetic materials which occurs in pair, of which they give about the transmission of this one, of which we will have to understand about many things here, about the dominance, of which about the dominance, we have various types of dominance. So we have dominant number one, so we're speaking about dominance, of which on the dominance we can have a complete dominance. We have incomplete dominance. Then we have the co-dominance. When we speak about the complete dominance, this is where one of the allele has to be dominant over the other. We can use in human being. Let's say one of our parents is tall, the other parent, that is made, let's say the male is tall, and our female is dwarf, that is short. The allele for the tallness is always dominant over the allele for the dwarfness. So it means all of our children will be tall because of their dominance of their father. So we are saying here, in terms an example, e.g., tall and dwarf. And on the tall and dwarf, the tall is dominant. So it means the offsprings, all the offsprings will be tall. We will do a genetic cross to confirm about the dominance of the tallness. When we speak about the complete dominance, it means none of the alleles is dominant over the other. So here none of allele is dominant over the other. None of the allele is dominant over the other. We can use an example of a flower. Let's say we have a white flower, then we have a red flower. This red and white flower, at the end, those are the flowers of the offspring. None of the flower will be having a red or a white flower, but we have a very different color away from the initial colors. So let's get, we have a pink flower. So we are saying here you have red flower as an example and white flower. What you get? You get pink flower. So that is a very different blended color away from the white and red color. So it means red is not dominant, white is not dominant over the red not dominant over the white not dominant over the red. But we have a very different color, which is a pink flower. That is where we are speaking about none of the alleles is dominant over the other. But for this complete, we are saying tall and dwarf. So we are saying the allele here, one of the allele has to be dominant over the other one, and the tall is the one we're calling what? Dominant over the other one. When we speak about the co-dominance, what are we speaking about? The alleles. It's about both of the alleles are expressed equally. So it's about both of alleles are expressed equally. An example, let's use the same red flower and white flower. What you get is a blend of both the red and white flower, of which now you get the red and white flower. What do we mean? This is what we mean. Red flower, white flower. But in the flower that we get is a blend of the red and the white flower. So these red and white flower now in the same one flower. So it's a blend of the color. None of these dominated the other. Both were expressed equally. So it means the red, white were both expressed on the flower. Let's use a genetic cross to confirm these ones. Let's do some genetic cross. I think we can do some genetic cross, and I'll start with the complete dominance. Let's do a complete dominance. Complete dominance. And I'm saying on the complete dominance, we are doing, we can use the Mendel's law. We do based on Mendel's, of which Mendel's used a pea plant. And when he was doing this pea plant, he studied various structures. He used the board, of which he used again the seed cord, and he used the height. Let's focus on the height, of which on the height this person did compare the planted shirt, that is the dwarf pea plant, with the tall pea plant. So we have tall pea plant and we have dwarf pea plant. Based on this one, these are something called parental phenotype. So we have parental phenotype. So we have parental phenotype, which is a tall pea plant and a dwarf pea plant. Then we'll have parental genotype. On the parental genotype now here, when we speak about the phenotype, what do we speak about? The phenotype are the external observable characteristics which will be determined based on the genes, which is the genotype, the genetic makeup of the organism. So the genotype determines about the behavior of the phenotype, but the phenotype is what you can see. You can see either being tall, you can see either being dwarf. But in terms of the genetic makeup, unless you go to the lab and you do some tests, so the parental phenotype, you will use the symbols, which will be T to represent the tall. But this one now, that is where I'm saying you will have something called homozygous and heterozygous. So let's say you have again T and T. This one is fully dominant, this one is not fully dominant. This homozygous tall, this heterozygous tall. But let's use the homozygous tall. So you have a genetic cross, it's a must you include your genetic cross. Then the dwarf, you use the small t and small t. Those are your parental genotype. Here we are using a genetic cross. You can be informed to use a Punnett square. So again, we have the gametes. On the gametes, now you just transfer these ones. T, you use the cycle. You do again another T. Then you have the small t and another t. Then now you do your fusion. On your fusion, you do this. It's from the first cycle to the first to the first on the other far end. Then again from the first, then you take to the second end. Then you take this one to the first. Then again you take this to the second. Hope you're understanding the genetic line cross. First circle with the first circle on the end. Then still the first end, then with the second. Then the second circle with the first, then the second to the zero. Then now you have your F1 genotype. We have F1 genotype. So it will be this T and the small t, capital T, small t. Still the capital T with a small t. You have the capital T with a small t. Then you have the capital T with a small t. So this one is about F1 phenotype. So you will have this one, you'll be asking about F1 phenotype ratio. Phenotype. Phenotype ratio is equal to what? And you can see all at all. Then you'll have the second one, which will be the F1 genotype. And the F1 genotype, you speak about all T. Let's understand this better. We are speaking about the genetics, of which we are speaking about the T, which is the tall, with the TT. You do your genetic cross. These that we are seeing here are, you can see it's F1 genotype. When we do our first planting, these are our parents. We are taking some seeds with some tall, we are taking other seeds with a dwarf. Then from this tall, you plant. These are what you get as your first harvest. That is your F1 genotype. And you can see in your field, all the plants were tall. Or you can plant the tall, you take the flower, that's the pollen, from a dwarf plant, you do some fertilization with the first, which is the tall. After the fertilization, what you get is all tall. Yet you use that dwarf. It's giving a good indicator that what the tall plant that it had been planted was an homozygous tall. Let's have a change. In case we use a heterozygous tall. In case you use a heterozygous tall, what will happen? So you're speaking about you're using heterozygous tall. We retain the first one. So you're saying you are using tall, but the tall now to be heterozygous. The first one was homozygous, so we are dealing with the tail heterozygous. So the heterozygous will be T capital with the T small. For the two dwarf, you will use the small t and small t. When you do to your gametes, we say first this T will be the first one, then this other one to be the second one. Then we have for the dwarf, which is the first small t and the second small t. You do your line cross, that is your fusion. You'll get this one to the first circle. You'll do this to the second circle. Then you'll have this one to this. Then the last one to that intersection. Then what you do, you have your the genotype. So it will be T capital, T small, T capital, T small, T small, T small, T small, T small. You can see we just have 2 of the tall, 2 of the dwarf. So our F1 phenotype, F1 phenotype ratio. You speak about 2 tall is to 2 dwarf, which will be 1 is to 1. Then F1 genotype ratio. What do we get there? It will be, now I said use the symbols. 2T is to 2 T small, which will be 1 is to 1 still. So when you speak about the genotypic ratio, you can see this one is some heterozygous tall. Then you have the dwarf, and you can see there are two. There are two. You can be asking based on percentage. How do you speak about the percentage level? This will be 2/4. So the tall will be 2/4, which gives you 50%. 50%. We are saying this direction. If it's percentage, it's 50% tall, 50% dwarf. So we are speaking about 50% tall, 50% dwarf. How do you get it? I'm saying it's about 1, 2, 3, and 4. So it's 2/4 times 100, 2/4 times 100. So you get 50%. The 50%, you get your 50 what? Percent. You get your 50%. You get your 50%. That is about the complete dominance. One has to be complete dominant over the other. That is, it has to be completely dominant over the other. And then Mendel used the ringed seed with a smooth seed. We can check about for the seed. That was for we are saying that was for what? That was for the height. And we are saying the height was either tall or dwarf. We are saying the second one, let's use maybe the seed cut. We are doing for the seed cut. And the seed cut can either be wrinkled, or smooth. So let's say Mendel used what? He planted wrinkled seeds with some smooth seeds. So your parental phenotype will be, we are saying wrinkled seeds with smooth seeds. What will happen? We are saying the wrinkled seeds with what? Smooth seeds. Then parental genotype will be what? We can make smooth seeds. We use S capital. For the wrinkled, we use small s. We are saying we use the wrinkled, small s. Then for the smooth, we use capital S. We have our genetic cross. So we have capital S. For the smooth, the wrinkled, we are using the what? Small s. Our gametes will be S small, S small. This other one is capital S. Capital S. How do you do it? This is how we do. Still our genetic crossings. Our genetic cross. Then we have the last one, which will be this. Then we do what? We have our F1 generation or genotype. Then we say we have here what? Fusion. You can see we will have the capital S. You cannot start with a small, so it will be capital S, small s, capital S, small s, capital S, small s, capital S, small s. So it means all are smooth. All smooth. Seed coat. What do we mean? When you speak about now all as having a smooth seed coat, it means that the smooth seed coat was dominant over the wrinkled seed coat. And that is what we're calling it, complete dominance. So next I'll take you through maybe about the bone color. Then we'll do this incomplete dominance and the codominance and even the sex-linked genes. Stay tuned, subscribe to get more information as we continue. Thank you very much.